So that's what was happening just in the nodal cell. Slow voltage gated sodium, fast voltage gated calcium, and ho-hum potassium voltage gated channel. Sodium rushing in, potassium rushing out, always does what it does. Sodium rushing in, depolarize, potassium going out, repolarize it. I don't have an extensive or an exhaustive sarcoplasmic reticulum and cisternae network here like I do with skeletal muscle. So this fast voltage gated calcium is going to pump calcium from the outside in. And that's going to help this cell to contract. It's going to do the same damn thing here as well. Okay, it's going to help these cardiac muscle cells contract, depolarize. Cardiac muscle cells contain shockingly leaky channels to maintain resting, which is minus 90 here. Minus 60 in nodal cells. I want them to fire easily, quickly, so they are minus 60 millivolts. Sodium, potassium, leaky channels, establishing resting. Calcium pumps. Calcium pumps are going to be all over the thing. Sodium, potassium pumps to reestablish as it dips slightly below. And nothing's different. Where I had slow voltage gated sodium channels in the nodal cells, you'll see here I have fast voltage gated sodium channels. Something that's commonly screwed up on an exam. I'm telling you now, it's already on it. So start looking at it. Where the nodal cells had fast voltage gated channels, now we have slow voltage gated calcium channels. And this is significant for what this thing does. And by this thing, I mean the cardiac muscle. By having this slow voltage gated calcium channel still working, it ensures that this heart contracts for a longer period of time. Again, what do they not have an extensive of? Sarcoplasmic reticulum and cisternae. They require calcium from being pumped from the outside of the cell to the inside of the cell. Extracellular calcium is a huge requirement for cardiac muscle. And we'll see that that slow voltage gated calcium channel ensures that nonsense. When it comes to cardiac muscle cells, these are the three events: depolarization, a plateau, and a repolarization. Okay, seems pretty easy. Depolarization, plateau, and a repolarization. Okay, and you can kind of see from the picture there what in the world is moving in and out. Pretty standard, pretty straightforward there. So let's look at these words. So here for cardiac muscle, the electrical events of the cardiac muscle, we have depolarization. What always starts depolarization? Sodium channels. Well, specifically here for cardiac muscle, it is a fast voltage gated sodium channel. This opens quickly, hence fast. Okay. And as you get, and it's spreading from one cell to the next, again, via gap junction. So this comes from the Purkinje fibers. Okay. Purkinje fibers brings that electricity into the cardiac muscle. And then quickly from one cardiac muscle cell to the next via gap junctions. And it's stimulating those fast voltage gated sodium channels. And as you can see, good God, these open really fast, really quickly, and boom, we go from minus 90 all the way up to plus 30. Bang, biscuit, really fast. As soon as we get to that plus 30, these sodium channels, these fast voltage gated sodium channels, immediately inactivate. Okay. They close up shop. They close up shop. As soon as we get to this plus 30 millivolts, okay, as soon as we get to that plus 30 millivolts, we get voltage gated potassium channels opening. That's going to help establish the repolarization event. But I also have slow voltage gated calcium channels open. These stay open for a very longer period of time. Again, fractions of a second, but it's still long. They are slow. They open up. Calcium comes across. Slow to close.
So do the potassium and the calcium open at the same time?
See, yes. It is, though, this that helps to bring in all that extracellular calcium. They both open at the same time, all right? But it's that huge influx of calcium that helps to maintain a longer state of contraction. Because what does muscle always ultimately need? It needs a stimulus, and it needs calcium, and it needs ATP, right? If I have all those things, skeletal muscle is going to contract. Well, here with cardiac muscle, ATP, yes. Okay. Stimulus, it just happened in step 1. But calcium here is coming from the extracellular. You get this huge influx of calcium. And that causes a much longer state of contraction. Now, is this going down? Yes. And that's what potassium is causing that thing to come down a little bit. It's not like skeletal muscle where we get up there and boom, it comes right down. Okay. In skeletal muscle, it goes all the way up and then boom, comes down. And then can go and then come down. But here in cardiac muscle, you get this huge plateau. And that's because of this calcium coming on in. This slow influx of calcium coming in does 2 things. One, it's extracellular calcium. So calcium binds to troponin. Tropomyosin moves, actin-myosin interact, yada, yada, yada. We get a contraction. It also stimulates the little bit of sarcoplasmic reticulum that we do have within the cardiac muscle to release its stores. Okay. There's no terminal cisternae here associated with cardiac muscle, but there is a small rudimentary sarcoplasmic reticulum. It stimulates it to, release its stores, as small as it is. So we have all of this calcium within the system, within the sarcoplasm. And what does that do? It interacts with troponin, and we get interaction between actin and myosin. This is what allows this cardiac muscle cell to remain depolarized. Why in the world would we want this thing to maintain a state of depolarization for just a little bit longer of a time, compared to skeletal muscle? We want to ensure that we pump as much blood effectively out of the ventricles as we can. So this slow voltage gated calcium channel opens up, brings calcium in, slow to close down. This helps us to bring calcium from the outside in. It allows us to maintain a much more longer state of contraction, depolarization, and the heart can be an effective pumper. That is the magic behind cardiac muscle. Okay. It can stay depolarized even if sodium has stopped coming in because of one thing: calcium. Eventually, voltage-gated calcium channels close. And it's not necessarily to know the thing. It's, around zero there. Okay. Voltage-gated calcium channels close. Potassium remains open. And we go back to our resting membrane potential. So the difference between what we just talked about with the nodal cells and what we talked about with the myocardium is this: nodal cells don't contract. Only myocardium contracts. Only cardiac muscle contracts. These nodal cells, AV node, SA node, AV node, and AV bundle, they are stimulated to contract at certain intervals by the brainstem. Okay. By sympathetic, by parasympathetic, and by a number of other inputs that are coming in that we'll talk about here. So these three guys, they establish the rate at which the heart is going to contract. And by heart, we're talking about the myocardium, cardiac muscle. As you can kind of see here, we have, and they don't do a very good job of showing you up here at the very top, there is an internodal pathway where electricity will travel across, okay, and synapse back onto that atrioventricular node. As these, think of them as cable wires. As these cable wires are traveling around through the myocardium, they will deliver the electricity to the houses, to the cardiac cell. That electricity enters the cardiac cell, and now it causes the calcium to come on in interaction between actin and myosin. And now the myocardium of the atria contract. At the atrioventricular node, slows down just a little bit, ensures those atria completely contract, push a little bit more blood down into the ventricles so they can completely fill. Allows them to continue to contract. That AV node slows it down just that much. Allows those two bottom chambers, those ventricles, to fill damn near completely. As the electricity moves from the atrioventricular node, you can kind of see it through this cable wire right here. And this region right here is my atrioventricular bundle. It now carries the electricity from the atria down into the ventricles collectively. Sends this electricity down the cables of the right and left bundle branch, traveling through that interventricular septum. We get down to the apex of the heart right down here. Those cables start to get branched and more numerous. Those are my Purkinje fibers. And Purkinje fibers are what deliver the electricity. They're the cables that come carrying the electricity to those individual houses, the cardiac cells. As they penetrate into the myocardium of the ventricles, they deliver that electricity to that myocardium, to that cardiac cell. And then from one cell to the next, just like we did from one skeletal muscle to the next, this electricity moves quickly from one cell to the next. via those gap junctions within them. As the electricity moves through that myocardium, it causes slow voltage gated calcium channels to open. Calcium comes on in, maintains a longer state of contraction for a fraction of a second longer than what skeletal muscle does, and then it relaxes. Okay. So it's the nodal cells, all this nodal tissue that I've been talking about, that is helping to get the electricity from the power plant, the SA node, and conducting it through cables to the other substations, atrioventricular node, atrioventricular bundle, bundle branches, Purkinje fibers, to deliver this electricity to every home in there, to every individual cardiac cell. Does that help a little bit? Yes. Myocardium is carrying out the contraction. It's the only ones that contract, you bet. Okay. APs, specifically the SA node. But if you don't have an SA node, the atrioventricular node can do it. Okay. If you lose the atrioventricular node and it's just atrioventricular bundle, then you can't, your heart isn't an effective pumper. But they set the pace. Okay. So those nodal cells set the pace for how fast the heart contracts and with how much force the heart contracts. Okay. And there are a number of inputs that come in and affect those nodal cells. Okay. We'll talk about those when we talk about affecting cardiac output here at the end. Okay. Does that help? Awesome. Very welcome. All right. All right. There's nothing new here, right? Let's see here. Calcium enters the sarcoplasm. Okay. From interstitial fluid, from extracellular. Interstitial is the stuff outside the cell, the space outside of the cell. All right. That's interstitial. So calcium enters the sarcoplasm from the exterior, from the interstitial fluid. And again, the sarcoplasmic reticulum, there's a little bit, not a lot. And the little bit of calcium that's in there gets released. Okay. Leading to contraction. Okay. Yeah. Binds to troponin. Yep. Tropomyosin moves. Actin and myosin interact. Yeah. Okay. Nothing. Okay. Calcium channel closes, and then I'm going to start actively pumping it out. And there is a whole fancy pants mechanism to it. You don't want to know about it. You don't care. Okay. It'll add an extra day to your life, and I don't want to do that to you. Okay. But there are some specific, pumpers out of getting rid of that calcium, right? I need to get calcium out, just like we have to do in skeletal muscle. Okay. So as soon as the voltage gated calcium channels close, we start actively pumping it all out. A little bit into the sarcoplasmic reticulum. That's what SR stands for. And then the rest gets pumped back out. As soon as calcium levels start to diminish, again, we relax, right? There's no interaction between actin and myosin, and there's no contracting. The hard thing about this, guys, is that we think of it like skeletal muscle. Muscle contracts and relaxes. Muscle contracts and relaxes. And that's not how this happens. I mean, this is fractions of a second where you're constantly contracting. Right? And it's hard to think in, 0.15 seconds. Okay. It's hard to think in that small time frame. All right. But that's what's happening here. And it doesn't really relax for a whole heck of a long time. I mean, 0.3 seconds, and then boom, we're doing it again. Because they have that plateau phase, I hope this makes sense that they have a very long refractory period. With skeletal muscle, I have an absolute and a relative refractory period. In the absolute refractory period associated with skeletal muscles, nothing can happen. In cardiac muscle, during the refractory period, nothing can happen. Okay. There is no contraction that can happen. There is no tetany. There is no technic contractions that can happen with cardiac muscle cells because of the plateau phase that it exhibits. Skeletal muscle, again, doesn't really have that. Okay. Depolarizes and repolarizes. Okay. And the muscle contracts. With cardiac muscle, I'm going to go over here, we have a much longer plateau phase than we do with skeletal muscle. Skeletal muscle does not have a plateau phase at all. Okay. With skeletal muscle, how do we maintain the state of contraction? By just increasing what? Motor units. Do I engage all my motor units when I start to contract? No. I increase the frequency of stimulating more and more motor units, more and more muscle fibers. And that's where we get that wave summation and tetany that can occur in skeletal muscles. Because of these slow voltage gated calcium channels opening up, I have a influx of calcium to help maintain that state of contraction. To help maintain that contraction of, I guess I should be specific, the ventricles. Because those are the pumpers of the heart, right? So this ventricle can contract for a fraction of a second longer because of that plateau phase. And it's because of that plateau phase, calcium, slow voltage gated calcium channels open, and potassium channels open, that allow this heart to not function at all in terms of being stimulated again to contract before it is completely relaxed, which is good. Because you wouldn't want your heart to go into spasm because of an increase in the number of impulses coming into it to contract. You want it to contract, completely relax, and contract again. And if it does not, then there's your heart attack event, okay, or your angina event, okay? It's not going to be an effective pumper. The body's going to try to compensate for that ineffectiveness. And that's going to lead to a whole host of issues affecting cardiac output. And we're going to talk about that in the last little bit of this lecture. Okay. We can measure all this activity. Okay. We can measure all this activity. All we do, strap the patient in, put a bunch of electrodes all over their body at specific locations, and we can measure how the electricity is moving through the heart. And that is what an EKG is, electrocardiogram. EKG, ECG, it's the same thing. Okay. This is how we can detect how electricity moves through the cardiac muscle. And there are a number of things associated with a EKG. The three things on this, you should know what they are. We have the P wave, the QRS complex, and the T wave. We have the P wave, the QRS complex, and then the T wave. Okay. The P wave reflects the changes, the electrical changes that are occurring in the atria, resulting in atrial depolarization. If you see depolarization, think contraction. Okay. So this is what represents the SA node, the atria contracting. This is the initiation of the SA node, and this little hump that exists here, okay, this little hump that exists here is telling us about atrial contraction. The QRS looks at the electricity that's happening through the ventricles. This is telling me when the ventricles are contracting. So the QRS complex is ventricular depolarization. This right here is telling me, this is what I see on that little sheet of paper printout when the ventricles contract. Again, depolarization, excuse me, depolarization is when we have contraction. P wave, atrial depolarization, QRS complex, ventricular depolarization. That little bump on the end, right there, okay, T wave. T wave is associated with the electrical changes that occur in the ventricles when it repolarizes. When you see repolarize, Frankie says relax. I have the P wave, atrial contraction, electricity, atrial depolarization. QRS complex tells me the electricity happening in the ventricles when it contracts. And the T wave is ventricular repolarization. So if we have atrial depolarization, ventricular repolarization, what's left?
Atrial repolarization?
Yep, the atrial is repolarization, right? Do the atria relax? Of course they do. When you contract, you got to relax, right? There is no other more contracting if you can't freaking relax. Where do the atria relax? The atria relax when the ventricles are contracting. Okay. And because the ventricles are much bigger, they generate a hell of a lot more electrical activity. And so they kind of get swallowed up. The atrial repolarization event gets swallowed up in that QRS complex. Now, with an abnormal EKG, you can see some extra little bumps and stuff like that. And we're not going to get into that because that's way above us right now. And you'll get it further on in your training anyway. If you see another little bitty bump there, that would tell you that there's some kind of electrical chaotic event occurring within the heart tissue. And then you got to figure out, why that is. There are a number of other things here, guys. I'm going to circle two of them here. I'm going to put an X through the two that I'm not going to have you worry about memorizing. But the the PQ segment there, it's associated with the atrial cells plateauing. The plateau is an important event associated with the heart cells only. So it is something that, you should know. So this PQ segment there, and it's just the space in between the P and the Q. Okay. This is telling me how long the atria are contracting for. And it's supposed to be at a certain interval. If it's extended PQ segment, that will tell you some things. If it's a very narrow PQ segment, that would tell you some things. ST segment associated with ventricular plateau. So again, they're kind of associated with the depolarization, that what the actual myocells are doing there. The intervals, they're important, but like the PR interval is basically telling you the entire time that the electricity is traveling through the entire conducting system, through all those nodal cells. So, that's what it tells you. The QT interval, how long, as you can kind of see, it's down there, the time the ventricular action potentials are occurring there. Depending upon your heart rate will depend on what your QT interval is. So if you have a very, if you have tachycardia, you're going to see a very irregular QT interval. So all of these kind of tell you something, but again, I don't want to get into it. P wave, T wave, QRS complex, and the two segments, those are the ones that I want you to know. Those are the ones that generally get asked about on exams and stuff like that. Know this one word up here for me, guys. Arrhythmias is just abnormal electrical activity within the heart. It could be many reasons why. It could be atrial fib, ventricular fib, a bunch of different things that we don't have time to go into. Third degree, first degree, second degree blocks. There's a whole bunch of the things that we could really get into that I don't really want to. No terminology, no words. Cardiac arrhythmia, cardiac heart, arrhythmia, abnormal electricity movement through the heart. That's what I want you to know. So that's the only thing you need to know on this slide, just that term, that name.